back to Science at the Source, a series from iCal, a P50 Center of Excellence at University of California at Irvine. That's a mouthful there. iCal stands for Impact of Cannabinoids Across the Lifespan. So today we are joined by Marciano Martinez, who is Marty. I'll go call her
Yeah.
today. Is a fifth year graduate student at UC Irvine, funded by a D-SPAN proposal or application. We'll get a little bit more into that in a moment, but we're so excited to chat with you about a really exciting review paper you put out recently about microglia and its role in addiction. So thank you for joining us, Marty.
Yeah, thanks for having me.
Awesome. So just gonna jump into it. So how did tell us about your academic journey, how it led to your study and your review of microglia in addiction?
Yeah, so I originally in undergrad, I was a psychology major. And in my senior year, I like didn't know what I wanted to do at all, but I had always had an interest in helping people with addiction. So I had talked to one of my professors, and he kind of said, well, like you can either do a master's or you can do a PhD. So I decided just to do a PhD.
Yeah.
Um, yeah. And I like didn't have any research experience. I graduated 2 quarters early. So in my last quarter of my senior year is when I decided this. So um, I like joined a couple of labs in my last like 2 quarters of my senior year. And then I applied for a psychology postbac because I thought I wanted to do a clinical psych PhD. And so I got into a psychology postbac at UCI, so here at UC Irvine for 1 year. And I was looking at how chronic cannabis users deal with stress. And so that was like a lot of human work. And I realized that I didn't really like working with humans that much. And so in my undergrad, I had worked with mice like briefly, and I knew that I liked working with the animal models. So I looked at other labs at UCI doing addiction work with rodents, and I saw uh, one lab who was doing adolescent nicotine and adulthood opioid stuff in rats. And so I reached out and I joined that lab. And so I did that also alongside the human work for like a post-bac period. And then I applied to graduate school and I got in here. And I started working with Steve. I was really interested in the adolescent THC stuff because previously I had done cannabis stuff in humans and also like that adolescent aspect in the other lab. And then just I think in my second year, Steve was like, "Oh, what are your thoughts about doing microglia work?" And I was hesitant, and like UCI has this huge AD neurodegeneration research group here. So in grad school, especially in the beginning, a lot of the like my cohort was like, "Yeah, microglia are so cool," and I was always like, "They can't beat that cool." um,
You were
and then I started to read. Yeah, I started reading about them and I was just like, oh, they're the coolest things ever. I'm obsessed with microglia. And I kind of just like really fell in love with microglia. It was like the first time reading scientific articles where I was like, this isn't boring and I'm interested. So I knew I had found like my thing. Um, yeah, and then it just kind of like went from there. Yeah, that's basically how I got into it. It's a lot of like chance, and I had just joined the right lab that was like looking into doing microglia stuff. And then luckily I was like, yeah, these are the greatest things ever. I want to do this.
Thank you so much for sharing that journey. I mean, it's really clear that you followed your passions and you tried a couple different things and were like, hey, I like the cannabis stuff. I don't like working with humans. No offense taken to me. But I like the animal stuff. And the link between like chronic cannabis use and stress is so interesting to me because a lot of people say that they use cannabis to help with stress. So, I mean, I want to hear more about that maybe a little bit later, but yeah, you've been saying a lot about microglia, which is kind of a hot topic right now. So before we jump into like how you got into microglia and addiction specifically, can you just define like what the heck microglia are first?
Yeah, sure. Okay. So microglia are the immune cells of the brain. They are really important for maintaining brain homeostasis. So disruptions like just pathogens, also like stress. Yeah, they're really important in like neurodegeneration stuff, cleaning up and like getting rid of plaques and tangles. And so, yeah, just overall, really important for maintaining brain homeostasis. And those things that can disrupt homeostasis can vary. So, like drugs of abuse are one of those things, also.
Yeah, I don't think a lot of people will realize that. I mean, this was new to me too, that we have like immune cells specifically that work in the brain. And a lot of people think automatically like your immune system is helpful when you're injured or if you're
Yeah.
sick with an illness. But what you just said is so fascinating that it actually like helps maintain kind of balance in the brain. And then if you use substances that might actually interfere with those systems. So walk us through like how you got from studying the immune system to addiction. Like, what's the connection between those really seemingly different fields?
Yeah. So I think I'm first and foremost an addiction researcher. So the microglia aspect just really came later on. I think that I think the two of them together made sense in my head because drugs of abuse are like this disruptor in the brain. Um, and yeah, and again, microglia are really important for like being the ones in the brain and being like, what's going on? We have to like fix this. And so even in the literature, we see that like drugs of abuse can affect microglia just morphologically, as well as like eliciting neuroinflammatory effects on them. And so microglia being immune cells and also like during developmentally sensitive periods, microglia are really important for me remodeling like brain circuitry and like plasticity, especially during these like really critical periods. And so I think drugs of abuse in adolescence, where the brain is just so sensitive and still undergoing a lot of changes, is a point in time where the brain is susceptible the most to these disruptions. And so, yeah, looking to see how microglia deal with drugs of abuse already in this sensitive period is something that I think is really important and should be looked at.
Absolutely. Yeah, and we were just talking in the prior episode, I chatted with your mentor, Dr. Steve Mahler, about adolescence, as you say, being like a super sensitive period when there's so much change happening in the brain, and microglia are doing a lot of that. So if you layer on these substances that we know changes those core developmental processes, I mean, that makes total sense that microglia would be involved in that. So thank you for laying that out. I loved your paper. So you published a paper recently called Potential Roles for Microglia in Drug Addiction, where you say that microglia might actually contribute to kind of the vulnerability to addiction or even progression into like something like a substance use disorder. So walk us through what were the key findings that you put together, and then if there was anything that surprised you in reviewing the literature on this topic.
Yeah. Um, Okay, so I think the way that we ended up organizing the review was really looking at the necessity of microglia within like stages or like components of addiction. So withdrawal, for example, is one where I wasn't surprised to see microglia being involved in because withdrawal can induce like a sickness feeling. And so since microglia are immune cells, it made sense that microglia would be involved in this withdrawal process. And so the studies looking at like depleting microglia so that they can show necessity of microglia within these withdrawal process saw that microglia were needed to have these withdrawal symptoms. So that wasn't as surprising, but there were other stages of addiction like dependence, where especially in one study, they saw that mice that were dependent on ethanol when they would voluntarily take ethanol, just like intake it, they see an escalation in mice that are dependent. And when you go ahead and deplete microglia and allow for repopulation with new microglia, you don't see that escalation. But this isn't true in non-dependent mice. So there's something about the mice already being in dependence that allows the microglia to be involved in this process that yeah, that makes them necessary for this escalation. And so I thought that was really cool and also a little annoying. I think like going into this, I thought, yes, of course, microglia are going to be involved because that's like my hope. But it's just so much more complicated than that. But I think the way that I think about it in my head now, after writing everything, is that microglia, again, are like immune cells. And so there are aspects of addiction where there's like heavy like stressors going on in the brain, I think. And so dependence is something that for sure is a stressor and that can elicit some type of immune response in the microglia. I don't know if that makes sense, but I think that there are certain aspects of addiction that aren't necessarily like that you would see with like acute administration or just like before escalation or dependence or any of that, where the microglia may not be as necessary for that. Yeah.
Yeah, no, that makes total sense. I mean, it's more complicated than just like microglia play a role in addiction. As you pointed out, there's so many different kind of aspects of addiction, and even thinking about like, okay, one acute or single drug administration, like you're going to get some sort of microglia response, but if you do repeated or chronic exposures, like that changes things. And
Yeah.
pointed out with withdrawal and dependence. I feel like you also need like a Team Microglia t-shirt, by the way. But you're right. I mean, there's so much more that we need to understand about its role in all these different addiction related processes too, and layering on the stress component as well.
Yeah.
Yeah, thank you. So you mentioned adolescence as being a really an interesting area or time period for you for the reasons we said previously. So what findings are you seeing in the adolescent space with addiction?
Yeah. Um, so not a lot. I think because the field of microglia and addiction is so new that the focus is really on microglia and addiction in kind of adult animals. Um, so because of that, there aren't a lot of people looking at adolescents. There are people looking at like prenatal exposure to drugs of abuse and then microglia, which I think is really cool and interesting. Uh, but yeah, on the adolescence front, there isn't that many, which I do think that there should be more people. Um, but yeah, the first one that I do know of that comes to mind is Kay Linker's paper from Francis Leslie's lab, also here at UCI. I think this paper is close to my heart because I was in a lab when that like was close to Leslie's lab while Kay was doing the study. So it was like my first introduction to microglia, even though I never would have expected to be doing microglia stuff. Um, but in her paper, they saw that there is with pretreatment of nicotine in I think it was like adolescent or juvenile rats pretreatment with nicotine led to subsequent acquisition of cocaine administration. But when they depleted microglia during this pretreatment of nicotine, they don't see that acquisition of cocaine. So, yeah, that's one of the only ones looking in the adolescent space, but a lot more in prenatal. And yeah, but I do think, I think with the field, there is like this increase in people now that are like really looking into addiction and microglia, which is really cool because I think a couple of years ago when I first started in this, there wasn't that much. And now I'm in a point where like every year there's more and more interest and more people doing it. So it like feels good to know that like this is the time period to be doing that type of work.
Absolutely. It's super cutting edge. And then, you talk about like, how things are so complicated when you layer on development. But yeah, things that happen in adults, we can't really extrapolate. So you need a new series of studies that look at different developmental windows, like the prenatal period, as you mentioned, where
Yeah.
we find totally different effects just because the ecosystem is completely different at those time periods. So
Yeah.
it's really exciting. One of the concepts in your paper you introduced or you talked about was innate immune memory in microglia when they respond to drug exposure. Can you walk us through what that means and how it might relate to addiction?
Yeah. So innate immune memory is where cells learn based on initial exposure to a threat. And then upon re-exposure to that same threat or similar threats, they exhibit either an enhanced response or an attenuated response. And so macrophages and monocytes have been shown to have this innate immune memory. It wasn't until like really recently that microglia have also been shown to have this. And so, um, yeah. So I think with drugs being able to elicit these inflammatory responses in microglia, um, you can it can lead to basically microglia later on having exposure to the same drug or a different drug and having a very different response. And so there is a paper by Piamelli, who is at UCI, and he had a paper in biological psychiatry looking at the effects of adolescent THC. And then they went ahead in adulthood, gave the mice a stressor, like an immune stressor, and they saw a downregulation of genes in the microglia that were important for the innate immune response. And so it seems that the microglia that had adolescent THC exposure, when they encounter this stressor in adulthood, they don't have that same response. And so I think that this can hint to kind of some vulnerability aspects, especially because some of the drugs of abuse, so alcohol, psychostimulants, and opioids, they act on the same receptors as certain bacterial like stressors. So LPS is the main one that was used in Piamelli's paper, but also used as like a stressor to elicit sickness in animals. And it acts on a specific receptor, this whole like 4 receptor that initiates this cascade of like inflammation that drugs of abuse can also act on. And so because of this, I think that there could be some hints of vulnerability that way with later drug use.
Yeah, and as you said, not just like to that specific substance, like if you're exposed to cannabis,
Yeah,
also later that could change how you respond to other drugs like alcohol and
yeah,
also other types of stressors like infections and stress itself. Is that what you're saying? That there's like this,
yeah,
yeah, that's
yeah,
really crazy to think about.
yeah,
Changes the way your whole immune system responds to these things.
yeah.
So you mentioned that things in adolescence might be different than adults. What are you thinking about the role of microglia in the adult versus the adolescent brain?
Yeah. So because in adolescence, there is this time period where your brain is undergoing a lot of rewiring, and microglia are really important for like remodeling synapses. There is within the field of microglia, the thought that microglia can prune synapses, although that is heavily debated. So I like want to just throw that in there. But the idea of microglia pruning synapses that are like no longer needed or need to be pruned away. Um, Either way, they're aiding in remodeling the brain circuitry. Whereas once our brain's already fully formed, the microglia in the adult brain just seem mostly to be, looking out for pathogens and anything that's going to disrupt that homeostasis. So it does seem that adolescent microglia have like a bigger role in remodeling and rewiring and like plasticity of the brain during adolescence. And so because of that, I think that disruptions to microglia during this period can lead to more vulnerability to drug use. And that can be either via direct disruptions to the microglia themselves or to processes that microglia are really involved in important during this rewiring.
Gotcha. Yeah, that makes total sense. And then you talk about a closely related concept of resetting, which I think gets at the idea that, if we use substances as an adolescent, are we kind of, in trouble the rest of our lives, or is there a way to kind of reset and recover from those effects? So talk about resetting.
Yeah, so this resetting aspect goes kind of with the innate immune memory aspect, but also that the aspect of drugs themselves impacting the processes during development that microglia are important for. So we can, in the case of innate immune memory, go ahead and kind of uh, deplete the microglia that have been exposed to that drug during adolescence or whatever time period. And then we can repopulate with new drug naive microglia that don't have that memory of that prior drug exposure. And so the idea here is that the way that the microglia would react then to a drug exposure, again, would be completely different and hopefully not as bad as it would be with that prior drug exposure. And then on the opposite side, where we go into like the remodeling and rewiring, um, we can imagine a scenario where microglia don't do what they're supposed to do. So say that they don't prune the synapses that they need to because the drug that was on board during adolescence has messed up that entire system. So we can go ahead and deplete those microglia and then allow for repopulation of new microglia. And then those new microglia are going to go in and go ahead and prune the synapses that they were supposed to. So it's kind of like reopening the adolescent time period. So yeah, those are the two ways that I think about resetting microglia.
That is really cool, and it sounds like super sci-fi, but I think it's really promising and could actually lead to new treatments for the addiction space. So super cool, and other spaces, I might add. That's amazing. I have a question about how microglia respond to different types of drugs, like THC versus opioids. Have you or others done any work or look into that area?
Yeah, a lot of the literature on opioids and addiction have seen that opioids elicit a very pro-inflammatory, um, yeah, pro-inflammatory response in microglia. So they'll release pro-inflammatory factors and cytokines. And then at least morphologically, a lot of the microglia display kind of like an activated phenotype where their processes are retracted and they're kind of big and angry looking. And for the most part, this is because opioids do act on that specific toll-like 4 receptor that elicits that immune response that like bacterial yeah, infections also bind to. With THC, um, there's not that much work that I've seen with THC in general. Just like in the adult brain, it can elicit morphologically at least different morphologies across brain regions. So in the cerebellum, the microglia can show up really angry, but in other brain regions like the hippocampus, they don't seem to be as angry. It also gets complicated because when you add things like adolescence, so how does like a drug exposure in adolescence affect the microglia? So adolescent THC by a couple looking at microglia morphology, and there's a couple of groups that have looked at this in the prefrontal cortex. They have always seen that the microglia are very or have like this pro-inflammatory microglia morphology or an activated phenotype. In my hands, I haven't seen an activated morphology. I see like no changes in morphology. But that could be just due to like the adolescent THC protocol because it does vary across different labs. Um, so that could be one thing. But I do know that microglia that are like in a dish, when you administer like endogenous cannabinoids and the microglia are already active, like activated, then it leads to like an anti-inflammatory effect. So there's like a lot of things going on where it depends on like the state of the microglia already, when the drug is given, what brain region, all of that matters with microglia. So it makes it very complicated.
Right.
Yeah.
Yeah, and then developmental stage of the person,
Yeah.
the animal. Yeah, super
Yeah.
complicated story. I like how you call them angry too, the way that they look at their face.
Yeah.
It's pretty funny. Thank you so much. I want to move a little bit more to kind of career development and next steps for you. So you've been so productive and gotten really prestigious awards and fellowships as a graduate student, like the NSF, GRFP. Acronyms are fun. And then recently the DSPAN F99K00 from NIH. Talk about the current, funding climate and how these opportunities have helped shape your career thus far and, what's happening now, if whatever you feel comfortable sharing.
Yeah, so I feel first and foremost really lucky and grateful to have received these grants, and they've really allowed me to do and pursue the research that I find interesting and that I'm passionate about. Um, yeah, I don't know how far Steve would have let me go down the microglia path if I didn't have my own funding. So I'm really grateful that this funding allowed me to kind of like carve my own little research path within my lab. Um, as for like the climate of everything going on, so the DSPAN F99K00 is a diversity grant. And things on that end are kind of uncertain. I think every day we hear from people in our cohort that are getting their grants terminated or not renewed. And so it's just a waiting game. It's more of like I know that it's not gonna be re-upped. And so yeah, every day I'm just like waiting for that email. But regardless of that, I still plan to like keep doing a postdoc. And yeah, I very much want to do a postdoc specifically in microglia because I am like an addiction researcher and I do want that microglia experience. So um yeah, it's impacted me in that it sucks. But like I'm trying not to let that be everything, I think.
Yeah, and that was Steve's message as well, is that it's not great, and we're really worried about, your peers who have already lost grants, will lose grants, maybe, aren't seeing such a bright and rosy future, and we don't, I don't blame them. It's really terrible right now. We're like actively telling scientists and people that wanted to go into science that we don't care about that, and that's really terrible because those are such important questions. So I commend you on all of your success so far. Like those are hugely prestigious and really difficult awards to get. So congratulations on obtaining those. And I really do hope and encourage you to continue forward and be resilient because what you're doing is so important and it really does have a lot of uh, promise for helping people who are suffering with addiction and other disorders. So thank you.
Yeah, thank you.
Yeah. So someone who's experienced a lot of kind of you have an interesting path. You said you didn't know much about microglia, and now you're, team microglia. So what challenges have you encountered, in kind of bridging these really different areas on your journey?
Yeah, I think because it wasn't like a thing when I like first got into it, there was a lot of skepticism from like actual microglia people where they're just like, oh, I didn't know microglia and addiction was a thing. So being met with like a lot of skepticism really, I don't know, sucked when I'm just like, no, this is a thing. It makes sense that they would be involved. But then I guess I just like I don't know, you just like read, which seems like such a lame answer, but yeah, you just read about it and you read more and more, and it like you just learn more. Um, and then yeah, it just makes sense in my head. And now like I I like was able to go to like a fully microglia conference last year. And that was spooky in its own way because these are like microglia researchers. They don't actually like, I mean, I don't want to say they don't care about addiction. It's not their like primary focus, but like my research was met with like a lot of interest. So that is like reassuring that I'm doing something that is cool and that they don't automatically just think is like not a thing. I don't know. I think it's also helpful that now there are more people doing it. So it is becoming something when it wasn't a couple years ago.
Right. You're really on the forefront, and there's a lot of pros and cons to it, but you're really carving out a unique pathway, which is fantastic. And I think that's how the coolest science happens, is you take like this body of literature in one area and then you apply it to a totally new space, and you're able to make really interesting and novel insights. So again, kudos to you.
Thanks.
Awesome. So last question for you. I want to give you kind of the last word. So what advice would you give for someone who's interested in studying these topics, other topics, neuroscience generally? What advice do you have?
Uh yeah, I I don't know. I feel like I'm so junior that I that I don't feel like I could give advice. But I think if I could give any advice, it would be just to, yeah, I don't know. Well, one, read. I think that that's the biggest piece of advice that I got throughout grad school. And that I think is really important. But also, like, if you're passionate about it, I think that you should do it and just, yeah, give it a shot. You don't know what's going to happen. And in regards to like addiction and microglia, I don't know. I hope we keep doing it and that something good comes out of it, and that hopefully we can eventually like help people with addiction because that's what this is all like why we're doing this, I guess. Yeah.
Beautiful. Thank you so much, Marty. I really appreciate your time. This was fascinating. And just a plug for some of our social media. If you are watching this on YouTube, we do have a YouTube channel, Science at the Source. So please hit subscribe. Sorry, shameless plug. And then I'll leave you one last word. And then on Blue Sky and Instagram and LinkedIn, we're opening our accounts, UC Cannabis or UCI Cannabis rather. So give us a follow there. And then make sure to just stay up to date with what the center is doing. There's going to be some really exciting work that's already coming out from the center, from our researchers. And then in the next couple of years, I'm confident we'll have more exciting discoveries that, will shape this space. So thank you again, Marty. Anything else you want to say? Last words?
Uh, no, I think I'm good. But yeah, thank you for having me. This was really cool.
Awesome. It was a ton of fun. Thanks again.